Hello once again, and we are in this daily reading exercise of from the book The Complete Guide Fossils and Fossils Collection. So there we go, and the chapter for today is early land plants. Before the evolution of primitive land plants, some 40 million years ago, when the land was rocky and barren except for mats of algae and mosses along the edge of the water, by about 400 million years ago, vascular plants such as rhineals and zoster phylates began to spread. These early plants reproduced by unleashing clouds of spores in the manner of simpler plants such as mosses and ferns today. One of the earliest and best known vascular land plants appeared late in the Silurian period. Cooksonia was a small and simple plant, yet it achieved worldwide distribution, although it is dominantly known as Eurasia and especially Great Britain. Its success may be largely due to its vascular system, which allowed water, minerals, and sugars to be distributed throughout its entire body. Cooksonia had no leaves like modern plants, but it was probably green and photosynthesized, captured the sun's light energy over its whole stem surface. It is recognizable by its characteristic smooth, wide-branching stem that ends in spore capsules shaped like kidney beans. Zosterophyllum, a late Silurian vascular plant, the Cooksonia group. Zosterophyllum is distinguished by having phyla-like and also edge-shaped branches, where each branch is divided into two equal stems. Each stem was smooth, and the spore capsules were clustered in the manner of flower spike toward the end of the stem, but on the sides, rarely the tip. Capsules split along the sides, unfurling like a fern to release their spores. Nutrients were shared between the stems by a network of underground root-like rhizomes. Plants like Zosterophyllum were probably ancestral to the giant lycopods or club mosses that dominated the cool swamps of the Carboniferous period. Goslingia. This fossilized remains of the early land plant show it to be the smooth stem with a distinguished Y-shaped branching pattern. The sporangia are scattered along the length of the stem on small-sized branches rather than clustered at or near the end. In addition, some stem tips are coiled much like those of a modern fern in a form known as circinate. Many of these plants were washed downstream by floods and deposited in oxygen-deficient mud. These anaerobic conditions prevented decay and allowed the plants to fossilize as pyrite or fool's gold, known as pyritosization, preserving in microscopic detail the cellular structure of the original plant as it was in life. Saudonia. Saudonia is distinctly different from other early land plants in being covered with saw-toothed spinous scale-like flaps of tissue along its stems. However, these flaps of tissue were not vascularized inwards, did not have fluid transporting pipe-like vessels, and therefore cannot be considered as true leaves. Their function is not clear. They may have been spiky defenses against early insect-like land animals. The presence on the flaps of stomata tiny holes allow the exchange of gases and water vapor between the inside of the plant and surrounding air, suggesting that they increase the photosynthetic surface area of the plant for greater capture of light energy. The sporangia of Saudonia are found laterally, and the branch tips are circinate, uncoiling like the head of a fern. So that was the early land plants, and I'll show you the picture. There you go. So that's all from my side today from the reading exercise. There you go. Bye.